Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes. Well, it's another day for the 11th class and we were doing the biological classification chapter number 2. In this chapter till now, since last video, we have completed the type of classification in which we have done the 6 kingdom classification. Moving on, once we have done it, since we have to study more about what R.H. Whitaker classification. So in this, the first classification, the first kingdom was Kingdom Monera. So this video would be just on Kingdom Monera in detail. Now, first thing you have to remember that Kingdom Monera has all what prokaryotes, means they are prokaryotic cell, characteristic feature. No other kingdom has a prokaryotic type of cell, only the Monera has. And they are on who are the member of this phylum, that is bacteria. Different types of bacteria are put into this. Okay, moving on, as we move further, then we have to study about the characteristics of the prokaryotes. Now the prokaryotic cell has what certain features present to it. Okay, so we're gonna be studying about those things. Just a minute. Okay, so before starting it, I am making a diagram of it. Okay, so this topic is structure of prokaryote. Okay, so there is what a body integrity is there, that is a cell membrane. Now the cell membrane has some outer growths, these kind of structures, certain bigger outer growth structures also present like this. Like this, and certain inner growth structures are also present. Okay, so let's just name that what we have done it. So this is what one such, so this is the plasma membrane. These outgrowth, the longer in size, okay, the bigger in size, they are what? They are the fimbriae. What are they? What they do, we will learn it over a period of time. The smaller one is pili. This bigger structure help in locomotion. So this is flagella. There might be cilia present. Okay, and this inward growth which you are seeing, this is mesosome. Okay, now around it there is presence of what? There is a presence of a liquid, slimy or a solid structure around it. Okay, I'm just making it like easier way. This structure which I just made, it is what? This is the glycocalyx. Okay, it is what? A protective covering. It could be capsular or it could be slime mold. Okay, moving on to the next thing. In between it, there is what? A rigid structure present. Rigid structure means which is not changing its position, and that thing is what? This thing is what? It is the cell wall made up of peptidoglycan, which we have already studied before. Now inside it there will be what? There will be presence of cytoplasm. Moving on, then there will be what? There will be nucleoid present, means the genetic material. So this is what? The nucleoid. There is presence of what? There is a presence of ribosomes into it. So ribosomes are there. Sometimes these ribosomes form chain like of structure called as polysomes. Okay, there on this structure, which structure? this structure there might be present what? Respiratory granules, because respiratory granules are associated with the plasma membrane. So these are what? Respiratory granules. Okay, moving on to the next thing, there might be presence of plasmids. What are they? We learn it later. Plasmids. Then there might be presence of chromatophores. So we have chromatophores or granules. This is the basic structure. Okay, let's just study about it. So there is what? There is a presence of cell wall. Okay, this cell wall is made up of peptidoglycan, or also called as murein, which is a type of mucopeptide. So this is a important for you to remember, which we have already discussed. Here we have studied what? That it is also what? Murein, called as murein. Then the cell membrane we have always remember it, it is made up of proteins and lipids. So it is made up of what? Lipoprotein. Okay, and there is a space between the plasma, like for if you see over here, like over here, this is the space which is present. So what is this space? This space is known as what? Periplasmic space. So this is the space between cell membrane and the plasma membrane is known as periplasmic space, and this space is analogous to the lysosomal functions, means it helps in digesting the complex substances. So this thing means, sorry, opposite to that, analogous means opposite to the lysosomes, because this space help in digestion of the complex substances. Okay, that is done. Then the cytoplasm is there. Cytoplasm is what? It is a fluid matrix. Okay, so it is what? It is the fluid matrix. Okay, which is present inside the plasma membrane, and in this all cell organelles are present. Okay, so the prokaryotic cells, the nucleus is indistinct. Inside it, if you see that all the organelles in the cytoplasm, it is indistinct, and because of that we call it what? Nucleoid. This name I have already told you. Other names which you have to remember, that it is called as incipient nucleus, gene pore, or fibrillar nucleus. You will remember these names, that incipient nucleus, gene pore, nucleoid, and the fibrillar nucleus. Now the nuclear membrane is absent. So nuclear membrane is absent. So it is not there. Plus it also lacks nucleolus. Nucleolus is what? It is a area, space present inside the nucleus. We will learn in the cell chapter. We will learn it much better. Okay. Now the prokaryotes also lack true chromosomes. Instead of false chromosomes kind of things are seen during their cell division, which is what? Circular naked DNA plus non-histone proteins are present. So prokaryotes do not contain the histone proteins but the non-histone proteins. That only you will remember, that it contains what? Double-stranded circular naked DNA with the non-histone proteins, which are what? Polyamines. This is an important statement. Okay. Moving on to the next, that the false chromosomes coils and form the nucleolar regions called as nucleoid. Okay, you will remember this fact. So no pure nucleus is there. Moving on, in the prokaryotes, the ribosomes are 70s. Okay, we will learn about them later on, but the ribosomes are important for what? For protein synthesis. Always remember. Now there are certain types of prokaryotes that are there, and we're gonna be studying one by one each of them. The first is what? Eukaryotes, Eubacteria. Okay, sorry, not the eukaryotes, it's the Eubacteria, which are 2 bacteria. Then actinomycetes, which you will learn more about in your second year of your MBBS. Blue-green algae is there in our syllabus. Archaebacteria is in our syllabus. Rickettsia you will learn more about in your microbiology, that is second year of MBBS. Mycoplasm, here we will study about it. There also you will study. And chlamydia, you will study about it here in this your 2 years of journey, more in your sexual reproduction chapter. Coming on the Eubacteria, that is what? The history. Who found it? Leon von Hook, who saw the first what? First living cell. Okay, so first living cells were these. Bacteria is also present in the pond area, and he told about this in his book. Okay, then there was this person F.J. Kohn and Aaron Burke who coined the term bacteria. Okay, Berger told that their bacteria is what Phosphita groups and wrote about that in books and more about their books. Then there are more people who keep on finding about it, and there was a person Robert Koch who found about the leprosy disease. Okay, and the leprae is the causative agent. Mycobacterium leprae is the causative agent. So these are what history about it. You don't need to know all about it. Just remember that Leeuwen von Hoek first able to find it. Okay, moving on to the next thing. The bacteria has different shapes. Okay, so what are the shape? Cocci shape means circular, rod shape, spiralan shape. They might have the flagella or cilia present, and the vibro shapes. That is a comma. Here the examples are given to you that you have to remember. So what are the example? Diplococcus pneumonia. So the name itself saying that Diplococcus. Okay, means coccus word is coming up. Okay, E. coli, that is Escherichia coli, Bacillus anthracis. Okay, about them, their diseases we will learn later. The Spirillum voltonis and Triptonema, which we will be learning Triptonema later on in the sexual reproduction of flowering, STDs, when we're going to be studying about in 12th class. Then Vibrio cholerae we're going to be studying more about in the 12th class. Okay, the next thing is that mortality of bacteria. This thing is not being taught by any institute. Okay, most of the institutes are not doing it, and on YouTube no one is doing it, but we're gonna do it. Everything which is being taught in the institutes, because institute level teaching should be at your place. Okay, so mortality of bacteria means that with the help of flagella or the cilia, how they are moving and how they looks like. So there are certain types how the arrangement of flagella is there. So let's just see that. So if atrochus means, A means absent. Whenever the word A is coming, A means what? Absent. So atrochus means these are those kind of bacteria where flagella is not present, like micrococci. How it will look? Like this. Circular. So this is how the micrococcus will look like. Then we have monotrichous. Mono word means what? One. That one flagella is present on one end. For example, this is the one bacteria body and one flagella is present like this, or this one flagella only. So this is what? Monococcus. So example, your Vibrio, Pseudomonas. So these examples you have to remember. So like this, for example, or better this way. Okay, or the bacteria which I made just before starting this topic. Okay, so that thing is what? Monotrochus. Amphitrochus. Amphi means what? Amphi means both. So means one flagella is present both the end, means here also and here also. Okay, so a single flagella is present on both the end like this. So this becomes what? Amphitrochus. Amphi word itself means both. In biology, try to learn with the help of what? By the term decoding the meaning of the words. That will be making much easier to you. Then Lophotrochus. And the Lopho word is taken for the bunch. So means here what happened? The bunch of flagella is present on both the ends. Like for example, if this is the one such bacteria, and like this. Lot of flagella present on both the bunches are present on both the ends. So this is what? Lophotrochus. Then Cephalotrochus. Cephalotrochus means it is what? There is what? A cephalo, what it says means head. So it feels like there's a one head and the body is attached to it. So means only one side there will be what? A bunch of flagella will be present. So cephalo means what? That head. So means what will happen? For example, this is the bacteria and this is bunch of flagella present to it. This is Cephalotrochus. Only one side it is present. Then Peritragus. Peri means everywhere. Okay, so how I will remember it? Peri seasoning. So peri seasoning, what? If you put the peri seasoning, every fries would be coated with the peri. Okay, so it's not like KFC which just put peri on the one side. Okay, it's like McDonald's peri peri fries, where we shake it and it filled every part of it. Okay, so what happened, this is the bacteria where the flagella is coming from any part. Okay, so what are the ones? Atrochus means A means absent, no flagella present. Monotrochus means one flagella present at any part of the body or the any end of the body. Then we have Amphi means both the side one flagella present. Then Loco means bunch of flagellas are present on both the sides. Then we have Cephalotrichus. Cephalo means only one side because head will be one side, other side would be the body, so that will be the tail. Peritropus means everywhere it is present, like Salmonella typhi, E. coli, they are coming into it. Okay, when they are not writing anything means that Salmonella, some species might be having, some might not be having this one. Okay, so this thing is there. Moving on, then this flagella which we are talking, it has a structure present. Okay, so first of all remember that the flagella has three parts. What are those three parts? We have basal body, hook and filament. So if we see over here, this is telling us about all of this. Okay, because in the diagram also when I'm making, I told you that there are certain structures coming out from the body. These are about plasma membrane extensions. So that's why we are going to be studying about this first. Okay, so let's just see over here. So this is what our flagella will look like. So this is the cell membrane. This is the cell wall, and in between this area is what? Pericellular space. So what happened in your cell membrane? What is happening? The cell membrane and the cell wall has what? The basal part of it. Okay, so if you see over here, this area and this area together, this is what? Basal part of it. Then comes this hook kind of thing, which is protruding out. It's joining the basal part in the cell wall and coming out, and from here the flagella coming out. This is the flagella or the filaments. Okay, so flagella has three parts. That is what? Basal part, there is a hook, and there is a filament. Let's just see about the basal part which is present. Now in this basal part there are certain rings present. There are two outer pair of rings, and there are one outer pair of ring, one inner pair of rings. Total 4 rings present, 2 outer, 2 inner, and we call it one outer ring pair, one inner pair. So if you see that this outer pair, the cell wall is considered to be the outer pair, and it has what? L and P ring. Can you see that these rings are there? So L and P ring is there, whereas on the cell membrane there is S and M ring. Okay, so there is what? L, P, S, M. Okay, so these thing you have to remember that there are what? 2 rings present, inner and outer. Outer means outside. Outside is what present? Cell wall or cell membrane? Cell wall. So means outer ring, pair of the ring will be present on the cell wall, and they are what? L, P. Okay, whereas inner pair is what's present on the inner side is what? Cell membrane. This is S, M. This you have to remember that SM and LP. Inner outer you can easily understand if you have understood that the outer would be the cell wall and inner would be the plasma membrane. Okay, this thing you have to remember. Okay, rest of the things if you don't remember it, no issue. How the arrangement is there, no issues. Okay, that is what flagellin proteins is present in the filament. That is the only thing. Okay, so flagellin is easy to remember. Flagellin means coming from the flagella. Flagella means filament. Okay, so this is what how the arrangement of flagella would be there. After flagella, the next thing is the pili. What is pili? Pili is also the extensions, and these pili are what? They help for the sexual reproduction in them. So what are they? Pili are smaller than the flagella, and they are more numerous, present throughout the structure. And if, for example, if this is a bacteria and this is the one pili, this is another pili of another bacteria. Okay, let's just see it like this. Okay, this is the one bacteria, this is my hand is one bacteria, and this is one pili. This is another bacteria, and this is this bacteria pili. Okay, how to do it? This bacteria pili. So what happened? This both pili join together and form a tube kind of structure. So this is called as conjugating tube. We will learn it. Now what happened? For this or this, anyone, whichever want, okay, which they will consider it, okay, they will give what? They give their genetic material, certain part of genetic material, which is what? Plasmid they give. Okay, we will learn it later. So what happened? The plasmid from this goes to this. Okay, so this is how what the genetic material transfer occurs. Okay, with the help of pili. So pili are what? There could be a longer pili or there could be a shorter pili. Longer pili is known as F pili. F is for the fertility or the sex pili. Okay, so there are what? These are numerous hair-like structures. They're smaller than flagella. Single is pili, plural is pili, singular is pilus. Okay, they could be long and shorter type. If the longer pili are there, they are known as F pili or the sex pili. Okay, and how they give the donor and all, we will learn about this in the reproduction topic. Okay, then the shorter pili are taking part in the attachments, and they often called as infective pili or the fimbriae. So here only we said that pili is a broad term, but fimbriae are what? These are the smaller smaller pili which help in attachment to the substrate, and this is present only in the pathogenic bacteria, commonly found in the pathogenic bacteria. Okay, so means they help in what? Like for example, this is the certain thing, like for example, this is the parts, for example, this is the bacteria and these are the pili. So they will do what? If this is thing, this can hold it like this, right? So this is the work of the pili. Okay, so the fimbriae one of it. Then they are made up of what? They are made up of piline proteins. Flagellin protein is made up of what? Filament. These are made up of piline proteins. Then there is a structure of the bacteria which we have already discussed, but if you want you can see over here, but here we're gonna be quickly going through everything. The first thing is what? That a Eubacteria has what? It has a cell envelope. Cell envelope means outer structure. So outermost thing would be what? Glycocalyx. Then there is a cell wall, and then there is a plasma membrane. So this is the three things given to you. Okay, now the outer glycocalyx. Okay, the glycocalyx is capsule or it could be slime layer. Slime layer means like very fluidy. Imagine it like that there is a what? Jelly present around itself. And capsule means solid. For example, this is what? A solid structure, like this solid wall kind of a thing can also be there. Now, so they have given that the slime layer would be what? It is a loose and thin layer, whereas capsule would be what? Thick and tough layer. Okay, form of what? Glycocalyx. Capsule is made up of polysaccharide and polypeptide, while slime mod is mainly made up of polysaccharide. Okay, so it helps the bacteria from the action of immunity of the host. Like for example, if a bacteria which has glycocalyx goes inside my body, then this thing will act like what? Armored. So glycocalyx is what? It is the armory protection things. Like for example, earlier what happened when the kings and the people go for the war, they have what? They have made up of all iron thing. Okay, so that it becomes like what? Capsule. Same thing over here. So who it is protecting? Who this glycocalyx protecting bacteria from? It is protecting it from the WBC. Okay, or the immunity. We can also call it immunity of the host. Next we have what? Cell wall. Always remember that cell wall give it a rigid character. Means rigid character means what? That wherever the cell wall will be present, a proper shape would be there, because the cell wall, like as it keep on depositing, the older cell wall keep on what? Dying off. Okay, so it is a rigid structure made up of peptidoglycan. We have to remember that. Now here the concept of gram positive and gram negative occurs. Gram positive, gram negative is what? We will learn about this characteristic later on. For now, just remember that there are two types of bacteria: gram positive, gram negative. Those who takes the gram staining, they are called as gram positive. Those who don't, they call it gram negative. What are the concept? We will learn it later on in this topic only. But for example, here you understand that the gram positive bacteria, their cell wall is single layered and it is thick. It is made of a peptidoglycan. Lipids are present in less quantity. These factors will come again also, but for now just remember that gram positive thing like gram staining is related to the cell walls. Okay, just remember that these organisms have single layered and they are thick, whereas gram negative bacteria, They are what? They are double layered. Their inner layer is thin, and it is also composed of peptidoglycan, but their lipid quantity is comparatively higher. Okay, so L form bacteria will be dissolved by lysosome enzyme when the bacterial okay, this don't we have to remember this L form at all. For now we can leave this point. Then comes the cell membrane. So cell membrane is what? Bacterial cell have their cell membrane, which is made up of lipoproteins like all the eukaryotic membranes, similar like that. Then comes the cytoplasm. Cytoplasm is what? In the bacterial cytoplasm, membrane bound organelles like mitochondria, other these things which you might have remembered from the 9th class topic, they are not present, they are absent. Different types of organelles are present in the cytoplasm of it. And bacterial cytoplasm do not move, like its cytoplasm is rigid. Normally in our cytoplasm, what happens? It's keep on moving like this, like water keep on flowing now. So the tides flowing can keep on happening. So imagine like that. But that streaming movement of the cytoplasm do not occur in case of the prokaryotic cell. So this is one of the major differences. Now the cytoplasmic organelles which are present, fast we're gonna see that. Okay, the first one is the mesosomes. What is the mesosome? It is inward membrane, plasma membrane growth. Okay, and it is what? It is also called as coronoid. Okay, so it is what innervates the cell membrane of the bacteria, invaginate into the cytoplasm at certain place and forms this. Okay, now the functions is like mitochondria. What is the function of mitochondria? It has a certain function related to respiration. So this is what it has help in the respiration. It also help in the cell division. It also help in the DNA divisions. Okay, so these things it help in. Okay, so here we gonna be writing it up that it help in cell respiration, cell wall secretion, it DNA replication, cell divisions. These are the functions. So just remember these functions. It will come back again later, but here only we are completing the prokaryotes. Okay, now there comes the storage granules, and or also called as inclusion bodies. So they have what? They have glycogen granules. Like glycogen is what? That is a food. So they store their food for their time of need. Then there is volatile granules. So glycogen is what? Carbohydrates. So it helps the carbohydrates or food. Whereas volatile granules, they are also called as metachromatic granules, and they have what? Phosphate, and function as a reservoir of the phosphate. Okay, so phosphate is also important for the formation of lot of organelles, which we will learn it biomolecules topic. Okay, then there is chromatin material, that is nucleoid. Nucleoid is what? It is also called as gene 4, inceptum nucleus or fibril nuclear. It do not have a cell membrane present. It is naked present like this. That area, it do not have nucleolus present. Okay, it is connected to the mesosome because then only the DNA replication time it will divide. Okay, so this thing is important, that nucleoid is connected to the mesosome because mesosomes help in what? Division. So this has to be divided, so it will also help in it. Okay, now the next thing is that certain smaller fragments of these DNA is also present, which is called as what? Plasmid. Plasmid gets certain extra features and they can replicate on their own. Means this whole DNA replication will be done by what? The nucleoid will be done with the help of mesosomes. But there are certain small fragments of DNA that is called as what? Plasmids. Okay, these are the smaller double-stranded DNA present in the bacterial cell. This is called as plasmid. Now the plasmids have the ability to replicate independently, and the plasmids are very important topic which will come later on. Lot of the teachers are not telling about these things in the 11th class, then the students get confused. But here we are learning everything which is needed even for the 12th class. Okay, so you can refer this for your boards also. Okay, so this concept we will learn it here only. What is that? That is a plasmids. Plasmids give certain extra features. They can divide on its own. So if the bacteria is dividing, they will divide in itself without the need of the mesosomes. Okay, why they do it, how they do it, we learn it later. Okay, so there are 2 or 3 different types of plasmids present. Like we have F plasmids or the fertility plasmids. Remember that I told you what after we done that this bacteria will have a pili which will go and attach with this bacteria pili, and they will transfer the material. So for example, if the material, like which material will be gone? Plasmid will go. This plasmid which has this fertility thing or the genetic material is called as what? It's called as F plasmid or the fertility factor plasmid. Okay, so this fertility factor plasmid has this, so this is called as F positive or the donor, because it is donating it things into this. Okay, whereas this thing is F negative or the acceptor or the female. So this is only they have written it over here. F positive is what? Which carries the F factors, means which is a donor and male. Okay, F negative cells means which lacks F factors, they are recipient by the donor and are called as F negative or the female. This thing you have to remember. Okay, next is the R factors. R factor gives the antibody resistance, means certain bacteria, if you eat the medicine, they will not die because they have a this kind of plasmid which protects them from that. Okay, so this thing is there, so just go through this. Okay, then comes the staining of bacteria. Now what is the staining of bacteria method is that when we have to study the bacteria or any organism, we have to stain it and then we can see it in the microscope, because or else it is what? Transparent. We can't see any transparent thing. Even the light is going through, it will pass through the transparency of it. So how do we see it? So we use the staining methods. So all these WBCs, RBCs, if WBC especially you want to see, platelets you want to see, you have to put them with certain dyes. Same, the bacteria also has to be dyed up. Now the dyed up concept is what was given by the H.C. Graham. So what he did, just see over here. So he did what? In the first step, he put the bacteria and pour what? On the bacteria he put crystal violet. So this is the bacteria. So he pour what? Crystal violet. Either crystal violet or genetin violet and plus Ki solution. That is what? Potassium iodide solution. And then he washed it. Okay, then he wash it after putting it, keeping it for certain few minutes. He wash it. Wash with acetone or ethyl alcohol. Okay, now after washing them, some of the bacteria retain this crystal violet color. They are not being washed off, and some do not. So those who retain the colors, okay, because this crystal violet will make them what? Violet in appearance. Look over here, I am making like this. This is what now they look like. This color is visible. Now after wash, two things can happen. One thing is what? The bacteria will keep on having this color, violet color, or they will become again transparent. If they have this, we call it gram positive. If not, we call it gram negative, because whatever the stain gram put, it has it, so gram positive. If not, we call it gram negative. So this is how we call it. Certain bacteria are also being stained with the help of other colors also, like saffron and all those things, which depends upon that what kind of bacteria they are. Okay, so gram positive and gram negative. Now here what happened? We will learn the features of gram positive and gram negative here only. So this is what it is. This is the difference between gram positive and gram negative one. So we're going to be learning about it. A gram positive bacteria will retain its what? Purple color. So it remains purple after washing. Okay, whereas gram negative do not retain the stain when washed with the alcohol or acetone. Alcohol or acetone, you know this. Alcohol or acetone. Now the gram positive, and earlier only we said that their cell wall is single, whereas gram negative is cell wall is double. Okay, so this is single layered, this is bilayer. Now the first one, it is made up of a peptoglycan, very less what is present? Your lipids. So this is made up of peptoglycan, this is also made of peptoglycan, but who was more thick? Positive was more thick, it was thin. So it was thick. You don't need to remember the values, but at least remember this, less thick, less compared to compared to gram positive. Okay, then the wall is smooth. Here the wall is wavy. Wall is wavy. It is smooth. Then it has murin content more. It has comparatively less, means peptidoglycan. This we have already been explaining the things. Okay, then the basal body of the flagellum has 2 rings only, S and M, where it has 4 rings because the wall is also bilayered. So of course 4 rings would be there. Okay, then mesosomes is quite prominent. Here the mesosome is less prominent. Okay, then a few pathogenic bacteria belongs to it. Now here you have to remember this, very few pathogenic bacteria belongs to this. Okay, whereas most of the pathogenic bacteria belongs to gram negative. Okay, so this is it. Gram negative and positive doesn't have to do anything with the glycocalyx level. Okay, the glycocalyx also help in that, so don't get confused over here. Okay, now teicholic acid is present in the cell wall, teicholic acid is absent. This can be asked to you because it was there in the PYQs, so we're gonna be studying about this. Okay, so remember these factors. One, if you remember, other is just opposite to it, not much difference to remember it. Here try to understand it and make a view of it, that what kind of bacteria will look like. Thicker wall, thinner wall, less basal, more basals. Make a diagram and remember it. If you're having problem in it, let me know, that I will make a short out of it. Okay, so coming back to the basic thing, that what is the nutrition in bacteria. Here comes a very important, how they are making food. Now the bacteria has, like, there are 2 types of nutrition. One is autotrophs, other is heterotrophs. Autotrophs means making their own food. Heterotrophs means they are dependent on other. In autotrophs, 2 options are there further. What are the 2 options? 2 options are either they are chemosynthetic autotrophs or they are photosynthetic autotrophs. Chemosynthetic means they are taking the chemicals and obtaining the energy from it. Photoautotrophs means they are using the sunlight, like the plants, to make the food. This is the autotrophs. What about the heterotrophs? In heterotrophs, either they could be the scavengers or the saprophytic, means dead decaying organisms. You know that what happened after, if you leave a dead body after a certain period of a time, it will be gone. All the flesh will be gone. Who is eating it up? It is being eaten up by the bacteria only. So the bacteria are what? Bacteria are the scavengers or the decomposers, or what's the saprophytes, dead decaying organisms. It is also acting like a parasitic. Parasitic means it go and infect the organisms. Like in our body, sometimes we say, no, this person has typhoid, so it is what? Bacterial diseases. Same thing like that. Okay, then there are certain bacteria which has what? They are free living also. They can sustain on the, like, other ways of eating the other things. Okay, so these are it. So this is only given to you. So here we're going to be learning one by one. What is that? That they show the extensive metabolic diversity. This is a very important statement from NCERT, that they are the most extensive metabolic diversity, because they are autotroph also, heterotrophs also, autotrophs types also, heterotypes types also. No other kingdom show this kind of variations. Okay, now in autotrophs, what happened? There are photosynthetic autotrophs and there is chemosynthetic autotrophs. In photosynthetic autotrophs, they are doing what? They are taking what? They are taking the energy from what? From the sunlight, in the presence of sunlight. So here there are certain examples given to you. What are they? These are what? Purple sulfur bacteria, green sulfur bacteria, purple non-sulfur bacteria. Here you have to remember that these sulfur bacteria are not using water as the source. Means a photosynthetic bacteria, there might be what? Carbon dioxide, water, and they are making what? Glucose. Okay, but these are the other bacteria which are the sulfur bacteria, and when the sulfur bacteria are there, they are using this as their source of, like, compound. Okay, not talking about the sunlight. Sunlight is being used, no doubt. Where they are, sunlight will be used up. So here the sunlight is there, this is sunlight, and here also the sunlight is there. The only difference is what? This thing. Okay, so this is the examples given to you. So 2 different ways are there. Even the compounds, they are basic compounds they are using, that can make a difference. Chemicals means they are using what? Nitrogen compounds to obtain it. So there are what? Nitrifying bacteria which oxidize nitrogenous compound to form the energy. Okay, so they are what? Nitrifying bacteria are the example of it. These two examples are given to you, which you have to remember: Nitrobacter, Nitrosomonas, and Nitrococcus. Okay, next is heterotrophs. In heterotrophs, they could be parasitic or there could be saprophytic. Here you have to remember two words, that is obligatory and the facultative. What is obligatory and facultative? Obligatory and facultative means if someone is saying obligatory means strictly. Okay, so here only I am writing what? That obligatory means strictly. So means they are what? They are strict saprophyte. Means they can't survive if they do not get the dead decaying organisms. Facultative means that they are saprophytic in nature, but if they do not get that, they can survive with the help of other organisms. Means they can turn into parasitic. Okay, so these are what? They can become parasitic. Okay, so they are normally what? They are normally saprophytic, but they can become parasitic. So facultative means if the choice is there, they will become. Obligatory saprophyte means what? Means they will only and only be dead decaying organisms. Okay, so this is saprophytic bacteria. Same goes with parasitic bacteria, that obligatory parasite means that 100% parasite means they can't survive on the dead decaying organism or any other way. They had to be what? These types of bacteria would be what? They will be parasitic only. Facultative saprophyte means what? They are parasitic. When the like here the question is that ma'am, why this parasite is written and then say that this bacteria is not parasite, it is saprophyte. Facultative means that if needed, then they could be parasite. Means normally they are not. Means normally they would be what? Saprophyte. What is left over. Okay, so this way you have to remember this. Okay. Then facultative saprophyte means what? They are parasitic, but if they are not able to do it, so they can survive in the dead decaying organism also. So that has been given to you. Okay. Then symbiotic bacteria. Symbiotic bacteria are what? These are an important example, because symbiotic bacteria are those bacteria which are living in harmony with the what? With the other. Okay, and they helping the nature in certain ways, like for example rhizobium, they help in the nitrogen fixation to the plants. Okay, so that is there. Then comes the respiration. It is also based on the facultative or the obligatory ways. Remember it over here. Okay, what I am saying. So there are aerobic bacteria or anaerobic bacteria. Aerobic bacteria means which requires oxygen. Anaerobic means do not requires oxygen. Now obligative aerobes. Obligatory means strictly aerobes, means they required oxygen for their survival. If I remove the oxygen or add anything else, they will die. Okay, so they are what? Azotobacter, complete aerobic and die in the absence of this. Whereas facultative anaerobes means what? They are aerobics. Okay, but if not present, oxygen is removed from the environment, still they will survive. Okay, normally aerobic bacteria, but can survive the absence of oxygen, like Acetobacter, Aceti, Clostridium, Titani. These bacteria names will keep on coming. Then obligatory anaerobe. Obligatory anaerobe means strictly anaerobe, means if the oxygen is there, they will die. Okay, so complete anaerobic bacteria and do not have a capacity for the aerobic respiration, like Clostridium, Botulinum. Whereas facultative anaerobes means what? They are anaerobic bacteria, but if the oxygen is present, then they can do what? Aerobic respiration. That means they have the capacity for the aerobic respiration, means fermenting bacteria like lactobacillus. Okay. Then comes the reproduction. Here we will understand it better, that there are what? There are two ways of reproduction, sexual and the asexual. In asexual, they show the binary fission. You know that one cell divided into two. So binary fission is what? This is the one cell. This bacteria will divide into what? Two different cells. So these are what? This is the parent and this is the daughter cell. So this is what? Your binary fission. What is endospore? Now endospore formation occurs during the unfavorable condition. What is unfavorable condition? Like for example, currently if this is I am a bacteria. For me the favorable condition is what? That there should be oxygen. Okay, there should be water. Okay, so that I can drink. There should be clothes, so that if I feel cold I can wear it and all those things. Okay, but imagine that what happened, water is not there. then I will die. But bacteria has a certain mechanism to survive. What it does, it reduces its metabolic activities, means whatever is happening, reactions, it will stop, because reaction requires energy. Remember, catabolic, anabolic reactions require energy, both are taking place in the body. So bacteria stop that, and it reduces its water, it dehydrated and make the, like, comes to the very closer places instead of taking space, and make a protective layer around itself. That different layers which it forms around itself, or the coating it's to, it protects from the hard environment, means if it's there. And when the suitable environment comes back, it open, it burst, multiply, or anything which will be learned over the period of a time, and comes out and regrow again. So this happen as the endospore formation, and here only they are showing you what different layers that is present. This is what, this is the bacteria. Now these are the different walls which you are seeing around itself. So there is what exoporynal basal layer, outer layer coat, inner layer coat, cortex, core wall, core membrane, or the cell membrane, which is this much, so small. Okay, so this thing is there, endospore. Then the genetic recombination, which is what, a type of a sexual reproduction. So their sexual reproduction is called as what, genetic recombination, sexual reproduction. Now in this, what happened, there is transformation, transduction, and conjugation. So there are three of them. So starting with the first one, that is what, that is your transformation. This transformation thing will be very important part in your genetic understanding for your 12th. So here only we are trying to understand what is transformation. In transformation, beta, okay, let me just put it like this. In transformation, what happened, that how do you transform? Okay, for example, I am a Varun. Okay, I am a Varun and you want to look like Varun. So what you will do? First you will get your head shaped, right? Of course you won't get your head shaped, but if you want to look like me, you will get your head shaped. Okay, your skin color, you will do makeup like this. You will put little 4-5 hairs as a so-called beard and mustache. Then you will wear a t-shirt like me. Then you wear a aviator style goggles, or maybe a aviator style specs. Okay, and you start teaching biology. So you transform yourself. How did you transform yourself? That by taking up my things. Okay, same thing happens. There are certain bacterias, and if take up the genetic material of other bacteria, so they transform themselves. Okay, so that thing happens. So transformation is what, that genetic material from one organism, or particularly is species or strain, is taken up by another, which change / transform the another or later strain. So this happens. For example, if this is a one bacteria and there is another bacteria which has a green color coat around itself. So what happened? The first bacteria which do not have this, it takes away this green color thing, green coat. So what will happen? This white color thing will change into what? Into this second one. The first is changing into second one over here. This is what? Transformation. Okay, we understand the basic thing. Let's just come on to the second one, that is transduction. Transduction occurs when the virus act as a mediator of genetic information, provider or giver or donor. This we will learn more about when we will be studying about the life cycle of the viruses. But here I am telling you just the basic things, that what happened. Transduction. Okay, where should I write? Okay, here. So the next one is the transduction. So in transduction, what happened? It is a part of giving the genetic material from virus to the bacteria. Okay, so it is what? viral genetic material to bacteria. Now here I am giving you basic understanding of it so that when the virus come, we can recapitulate it in the bigger way. So what happened if, for example, this is a bacteria. For example, I am a bacteria, okay, and the virus want to attack me. Now viruses attack humans also. Viruses attack like coronavirus, right? So viruses attack animals, plants, and bacteria also. Everyone, like everyone they can attack. So they are smaller than that, okay? So now what happens that, for example, if a virus will come like this, attach itself. I'm giving one such example. And what the virus will do, this virus has a genetic material, it will push it inside my body, its genetic material, and then it will leave its place. Okay, genetic material come inside my body. Now that genetic material will do what? It will go and join with the DNA of my body, which is present in every cell. Okay, so it will go inside every cell DNA, means whatever the amount it is, and get incorporated with the DNA of my body. Okay, and then it will say to my DNA of the body that stop doing all the function, make my copy. Okay, means replicate me, make more DNA. So it makes DNA and the virus. So more virus will keep on forming. Means me will become what? My cells will becomes the host or the factory of producing what? To the more viruses. Understood? This is transduction. So here only I am said that when the virus that goes and attack and it do what? It do the changes. This method is also being employed over here at the genetic recombination. We will learn in the virus topic over here. Okay, so transduction is certain genetic material is being provided from one to another. So transduction is what? Genetic material by this way also can be transferred and they can make a new species of the things. Okay, and the last one is the conjugation. Conjugation, remember just now I talked to you about what pili thing. So that is what? The conjugation. Okay, and once the pili will join, this is called as what? Conjugation tube, positive to negative, male to female. Okay, that thing will happen. So here this diagram, if you see, is telling you about that conjugation only. So this is the F1, means it has a F plasmid, it do not have to F negative. Okay, positive means plus, negative means not present. Then what will happen? Conjugation tube forms with the joining of pili, and from this thing, it is present over here, it goes inside to this one. Now this becomes what? Positive, this becomes negative. Okay, so it becomes what? This forms the positive ones, but you know that there might be a multiple copies of this present, so it will replicate and form another copy. So now both will be positive only. If they are not making copies, there are certain plasmids which can't make their own copies, but for now we are saying that they can make their own copies, so it can becomes F negative also. Okay, so here it changes and the new features has been added to it. I hope this is understood. Next thing is the examples which you have to remember, and then we go with the classifications. So economical importance bacteria, which bacteria is useful to us? Common example, curd. Okay, so we have to study about the harmful and the bad ones. First is given what? First are given to you is the harmful ones. So disease causing bacteria, we will be learning more about in the 12th class, but here we will understand something. So if in your exam, and if you for like the unit test or FAs or whatever the exams that will be there in your school internal assessment, so these questions can be asked into the MCQ by your school teachers, that to show that the paper is so tough level, but we have already covering it up, right? So let's see that tuberculosis, it is caused by Mycobacterium tuberculosis. Okay, so like when you are writing it individually, you have to underline it. It is easier to remember because tuberculosis is the disease, TB, and tuberculosis is the species name. Only the Mycobacterium you have to remember. Next is leprosy, which is by Koch. Okay, Robert Koch, this person was the one who find it up. Okay, so leprosy, lepre, and this is also by the Mycobacterium only. So Mycobacterium we have studied one time genus, another species in the same genus has been doing the leprosy. Then tetanus Clostridium tetani. So here the name is tetani, easy to remember. Only Clostridium you have to remember. Then typhoid typhi species. Which one? Salmonella. Only Salmonella you have to study. So Salmonella typhi. How you will remember it? Write it, recite it every day. Then only you will remember these things, or you will forget. And these are all important for the NEET point of view. Don't think that it's not given in NCERT or something. It is given in NCERT, but we are combining it and learning it at this point of a time. Okay, so that later it becomes easier for you, because right now we are in the more in the kind of like pump up to do something to beat the NEET in 21, but this thing will keep on going up and down throughout the journey. But you stay with me, I make sure that all of you who are there, they get selected. Personal guidance sessions will also be given throughout this journey. Just be the part of this journey. Okay, let's just see that the cholera, Vibrio cholerae. Vibrio, how it looks like. Tell me, comma shape. Okay, this is what we have studied. Moving on to the disease in animals, only 2 disease. If just one disease also you will remember, that is fine. That is anthrax. Now the syllabus is not there, but we will study it. So anthrax is done by anthracis. Easy to remember. Same name is coming, species Bacillus. The Bacillus anthracis only you have to remember. Okay, so easier to for you to find out. Then black lag happens in animals, that is Clostridium chavini. If you don't remember this, that is also fine, but anthrax is very important. Then disease in plant. First is citrus cancer. Citrus cancer means citri. Citrus word is for the fruit. Okay, citrus fruit like orange, lemon, all of these. So these are what citri is the bacteria name doing what? Citrus cancer. Or this is by the genus Xanthomonas. Then crown gall in many plants. This is Agrobacterium tunificans. This remember, Agrobacterium tunificans. These tunificans, they will come up again in 12th class. So here you have to remember that what it happens, it causes tumor-like things. Okay, in the plant roots. Okay, so that tumor is what? Crown gall. Okay, then black leg and the soft rot of potato, that is Irvina Caratovora Athrosiptia. Although you don't have to remember this one. If you don't remember, no issues. Okay, this you can leave it if you want. Okay, so this one might not be asked to you ever, so you can simply leave this one and this one. Okay, moving on to the next one. This can be asked, that the bacteria leaf blight of rice. Okay, this is done by Xanthomonas oryza. So oryza word is used for rice. Remember it. So oryza is there. Xanthomonas citra, Xanthomonas another, oryza is for the rice. Okay, one is for the citrus fruit. Then the denitrification or denitrifying. Sometimes what happen in the soil, the nitrogen content is reduced. You know that nitrogen is super important, and plant get nitrogens from the soil. Okay, now just imagine that if there is a bacteria which is living, which is using nitrogen as a source of making food. Remember, I told you what? There are there are chemical synthesizing bacteria, which are what? Nitrobacter. Remember, Pseudomonas or Nitrosomonas. These were examples we have done now. Go back in the video and check again. So those bacteria will do what? They convert the soil nitrates into nitrite and nitrogen. And nitrogen, it is what? If it is in the form of gas, it will leave the soil and it will go into the air. Right? So this thing will do what? It reduces the fertility of the soil. So these are the bacteria. Theobacillus denitrification, with removing the nitrogen content. Pseudomonas denitrifying. These are the bacteria. So remember that these bacteria, Nitrobacter, Pseudomonas, Nitrococcus, Nitrosomonas, these are the one which is adding the, like, utilizing these things. So these Pseudomonas is one such example. Theobacillus denitrification. They're reducing the nitrogen content, means they are spoiling the soil. Okay, moving on, food poisoning. That is what? Botulism we call it, done by Clostridium botulinum. Remember that this bacteria survive in anaerobic conditions. So examples are meeting here and there. So it is a lethal type of food poisoning, and it can survive without oxygen. Here also it is, remember. So it can survive without oxygen. Means what? They are not obligatory. Means survive without oxygen means they are facultative anaerobes. Means when they need it, they can be anaerobes. Okay, just remember this fact. If you do not understand this thing, comment it down. I will make a short out of it to make you understand this thing. Okay, coming up to this, they can cause the death also, paralysis also, and all those things. Water pollution. Okay, so bacteria causes water pollution. We know that. Example, Vibrio cholera causes the diarrhea and all the Salmonella typhi called typhoid-like fever if we take the contaminated water. Then biological weapons. Biological weapons are what? Remember, you might have heard about it during the time of COVID that coronavirus is a biological weapon made by China, and throughout the world it was thrown to reduce the economy or everything, right? That is what. So what all other countries do, they make certain bacteria, viruses which affect the population, okay, which reduce the country's economy, growth, prosperity by doing this kind of attacks. So these bacteria act like a biological weapons and utilized in that way. Like anthrax means, if for example, if I'm a China, take an example, and I made the anthrax virus and put it inside the poultry farms or the cattle harvesting area. So what will happen? All my cattle will be soon infected with the anthrax. And if that will happen, what will happen? My milk and my meat that is being produced, that industry will go down, and it will show the effects on the people also. Economical burden would be there, health crisis would be there, and what not. Okay, so this botulinum, which is what? Lethal, dead, it can cause death also. This is also can act like bioweapons. Cholera causing bacteria can cause diseases and those things. So these are the problematic situation with the harmful effects of this. Let's go with the friend effects of it. That is what? The useful effect of the bacteria. Like ammonifying bacteria, they do what? They convert the proteins into ammonia. Where the proteins are present, as we keep on learning this thing now, you will understand that our body major all works are being done by the proteins. What does that means? That, sir, if I'm able to speak, it's because the proteins are forming for this function to occur. Understood? Means I'm able to do it because the proteins are forming who are making me do that. Okay, so everything is being done at the molecular level with the help of proteins. So when I will die, what will happen? Proteins are present in my body. Proteins are made up of ammonia, which is the basic units, which has a nitrogen, which is a useful compound for the organisms. So they do that Bacillus bulgarius will convert the protein in the dead decaying organism into ammonia, which could be utilized by the plants or the organisms for their growth. Okay, then nitrifying or nitrifications, opposite of denitrifying and denitrifications. So this will work over here. This we have already discussed before also in this topic. Moving on to the next thing, that is nitrogen fixation or nitrogen fixing bacteria. Means, for example, nitrogen is present in the soil, but is the plant is able to fix it? Is the plant is able to take it? Maybe no. So there are certain bacteria which are working. So some which are living in symbiotic relationships. Symbiotic relationships means that living like a roommates. So roommates, what happened? Roommates, one person will make the food, other person will wash the dishes. This happens same like this. So here what will happen? These bacteria which changes the nitrogen, they uptake the nitrogen, give it to the plant in return gives it to the certain shelter or maybe certain food or whatever it is. So these such examples are given to you, which will be asked to you in the question papers in the 12th class. So what are the examples which are of symbiotic relationship? Rhizobium, which is present in the root nodule of leguminous plants. Means these leguminous plants has what? They have a nodule present, like for example in the roots. For example, this is a root. So in this plant, what happened? That they are present like this as a nodule form. So these are the nodules. So we call it, these are the present in the roots. So root nodules. Which plant it is seen? The Leguminosae family or the legumes plant. It is seen. Then the next example that is given to you, that as Rhizobium, which is present in the stem nodule of the Sesbania plant. So in the stem also it is present. So Rhizobium. This is also Rhizobium. Other one is also a simple rhizobium is for the root. So R for this, the root. Azo comes means upper than the roots. Remember. Then azospiralium, they are on the root surface of the cereal plants. So this is also important. Way. Frankia, these are the filamentous and they are the type of actinomycetes. What are they? We will learn later on. They are present in the root nodules of non-leguminous plants. You don't have to remember the two examples, but at least you have to remember what? You have to remember this much. Okay, so the next thing after this is what? Asymbiotic relationship means they are free living in the soil. They don't require to make an association with the plants. Like for example, Clostridium, Azotobacter, Azilospirolium, Berzernickia, Rhodospirolium. These examples you have to remember. So what example you have to remember? Clostridium, Azotobacter, Azilobalium, Berzernickia, and Rhodospirolium, because these are given in your NCERT somewhere here and there. So this, at least remember it. Okay, so the next thing is what? The next thing is the dairy products. We know that milk is converted into curd with the help of Streptococcus lacti or the Lactobacillus lacti. So it increases what? When it is milk is converted into curd, it increases the B12 level of the curd, which help in what? It checks the disease causing bacteria. Then we have antibiotics. In antibiotics, what happened? Like your streptomycin, amoxicillin, tetracycline, all of these things has been made. The first antibiotic is the penicillin, and this was found by Alexander Fleming, which we will learn more about later. On the industrial levels, we form vinegar with the help of Acetobacter aceti. There are retting of fibers. Retting of fibers means that plant fibers from the plants, we obtain the fibers with the help of bacteria like Clostridium, butyric acid bacteria. These things are there. Then curling of tea or the tobacco leaves means adding the flavors into it with the help of bacteria is also being done. Then production of vitamins, different types of vitamins can be formed with that. E. coli has certain vitamins, Propionibacterium, vitamin 12, Clostridium botulinum, vitamin 2, riboflavin. These things would be there. Then purity of Ganga water is because of the presence of a large scale bacteria into it, that is Bacterovirus. Okay, so this is an important one because we say that Ganga water can clean as much as thing because of the presence of this bacteria into it. Okay, then pollution indicating bacteria are also there, like E. coli presence will tell that it is highly polluted. Okay, so this thing is also there. Bacteria for the genetic engineering like Agrobacterium tunificans, I have already told you about that. We're going to be learning about it. The disease also, we have seen that one variety of it causes disease also, that is what? The tumor formations. Okay, now we're going to be studying certain of these species, okay, in detail. Okay, starting with the blue-green algae. Now in the two system classifications, they were included into the plant kingdoms, okay, but now they are in the Monera, okay, and because they are prokaryotic. Now they are often referred to as what? Cyanobacteria. Cyano means because of the color. Cyano is a blue color thing, blue greenish color state. And International Code for Nomenclature of Bacteria has suggested this thing. So we have already studied this ICNB. Okay, I want to make it clear that in that video I might have written it IC back N. Okay, that is not back end, that is ICNB. I have come like I'm here on video also. I'm telling you that there was a mistake over there, and here and I have like made it correction in the comment box. Okay, so do check that out. Keep a check on that also. Okay, so BGA is a fresh water mostly, but it can be present on terrestrial environment or the marine water also. Now there is what? There are pigments present, and it is believed that they are initially the photosynthetic bacteria. If you remember that when I was doing the classification in the last class, I was telling you that first this come, then this come, then this come. So I told you that first was the heterotrophs, then the chemoautotrophs, and then the photoautotrophs. These were probably the first photoautotropic organisms that were formed, that is cyanobacteria. So that was why that is here a very important statement written, that is that they are the first organism that produce oxygen on the planet Earth. And they have what? They have chlorophyll A, chlorocarotenoid, phycocyanin, cyanin is for the blue, and the phycoerythrin, erythrin means red color present to it. And if the red color is present, means they are not essentially always blue-green algae. They could be red in color, and one such example is what? This Trichodesmium. So Trichodesmium is red in color, okay, because of presence of what? High amount of phycoerythrin pigments, okay, and it causes what? it makes the Red Sea red in color. And it is due to this cyanobacteria, which is also considered as blue-green algae, but in reality they are not algae. Algae are what? Algae are the plant kingdom. Okay, the first division. Tresmodesm, very important, you have to remember it. Now there are different forms of it. They could be unicellular, like spirulina. They could, which is a rich source of food. Okay, for astronauts they have been used. For cattle they have been used. Then colonial form like Anabina. Microcystis, they occur in the colony form, and filamentous is long form like trichome, okay, like Osteoculateria. So these examples you have to remember that they could be unicellular, they could be in the colonies, they can't be long filamentous form. Okay, then structure, if you have to study that, they are gram negative bacteria, and if they are the gram negative bacteria means they will not take the stain, means they have somewhat thick bi-layer thin layers. Okay, so they don't seep down deep into it, so everything will come over here on the basis of gram negative features which you can write into it. So once you learn about it and once you know that which bacteria is positive negative, you can write the next features. So this is only being explained to you over here. Then their cytoplasm is divided into peripheral cytoplasm and the central cytoplasm. Here I want to show you the example. If you see, so this is what this area and this area, there are this is a central cytoplasm, this is a peripheral cytoplasm. Okay, so if you see that in the central cytoplasm there is what? Nucleoid present, ribosomes present. Okay, that's it. So means it is what? Center part is little distinguished because where the genetic material is working over here, whereas outside area where all the other features of the bacteria are there. What are the features? That there will be present of what? Ribosomes present there. There will be present of granules. So these are the granules which might be containing either glycogen, sulfur containing, whatever it is. Then there is what? There is this cytoplasm, most of them. Then there is this cell, thylakoids are there. Thylakoids are what? These are food forming structures, thylakoids. Okay, so these are what? Photosynthetic pigments are present inside this thylakoid. So here only I am writing it. So these are what? This is the photosynthetic apparatus. Photosynthetic apparatus. Okay, so thylakoid and inside it presence of pigments will help in it. Okay, so this thing is there. Then coming up to the next thing, these thylakoids are also called as chromatophores. I have already told you there are chromatophores present in the bacteria's prokaryotic cells. So that is thing has been written over here also. That's why I'm not repeating the things which are written over here. I'm explaining most of the things, and then over here all the things has been written in the notes. These notes would be available on the my Telegram channel, which links would be in the description, as well as these will be available on the WhatsApp channel link also. That link is also in my description. Okay, so these things are there. Then there is what? That nitrogen fixations. Okay, this thing is understood. Symbiotic and all this I have explained to you before, that BC, like blue green algae, could be symbiotic form or the free living form. Examples has been given to you, which you have to just remember it. Now the important point is over here how they look like. If they are the filamentous form, that is nostoc, which is given in your NCERT, which you have to definitely do. So in the nostoc, if you see that it has a special type of cell called as heterocyst, which is what? It is a thick walled, it is non-green part. Then what is its role? Its role is the nitrogen fixation. Nitrogen fixation, because the enzymes which work for the nitrogen fixation can't work in the condition where oxygen would be present. Okay, so means certain problems would occur. Just remember that. So that's why they make a thick wall so that oxygen can't come inside it. They can simply fix the nitrogen. So nostoc is there. Nostoc, Anabena, these things we have put it into the paddy fields. Paddy fields is what? Rice fields. So that over there they can add what? Nitrogen into the soil so that the rice can grow. Then the reproduction coming on the vegetative and the asexual. Now question is, what is the difference between vegetative and asexual? Think about it. Well, in the vegetative reproduction, what is happening that a certain body part is working. Okay, so certain body part means like binary fission, we have the binary fission, we have fragmentation, where what is happening? The cell is dividing, fragmentation, part is dividing. Okay, this is the vegetative reproduction. What is asexual reproduction? Asexual reproduction means it occurs during unfavorable conditions. Vegetative reproduction is occurring anytime. Okay, when the unfavorable conditions, certain spore kind of things forms. So asexual reproduction has something to do with the spores, means special structures present. How I will remember it? Like your sexual reproductions, when the gametes are being formed, special structures. Here, asexual reproduction, special spores will be forming up. So till now, where we used to just indiscriminately in 10th class write that asexual reproduction, vegetative reproduction is same thing. No, beta. Vegetative means the body has two different types of structures or cells present. Vegetative cells or the reproductive cells. The vegetative cells will do what, beta? That your, for example, in the plant, your leaf, stem, roots, they are vegetative cells. If anything is occurring from this part, new plant is forming from this part, is the vegetative reproduction. Asexual reproduction means special spores will be forming inside the body. Particular structure will be there for forming these things. Okay, so that is the asexual reproduction, specialized thing. So here that happen in the unfavorable conditions. So it is a protection to overcome the unfavorable conditions. So what happened? Aconites formation will occur. This is very important. That in the nostoc, one another type of cell will form, that is aconites, which will what? They will form multiple, like they will help in tide over the unfavorable conditions. Okay, then water blooms. What happens? Sometimes this blue-green algae occur too much into the area, into the pond, for example, in the marine environment, it will relate. So what will happen in the pond? It appears green in color. It takes away all the oxygens, all the life form which is present, it will be eaten up, and it causes what we call it, what? Water bloom. Okay, so remember it. So this bacteria is not very good. If it grows too much in the amount, it takes away all the life forms. So it is destructive in nature. Okay, so this, because it releases certain toxins which will delete the, or which will delete, or basically it makes a death of the organisms which is present over there. So this thing is there, but it is good also, bad also. Okay, so these activities, economical activities are given to you. Next is the mycoplasm. Now the mycoplasm, first thing you have to remember that they are the first, they are having other names also. What are the other names? PPLO. What is PPLO? Fluoro pneumonia like organisms. Means they are those organisms which will cause pneumonia. Okay, so these things are very small. They are the smallest cell, because after that nothing is more smaller than mycoplasm. They do not have cell wall, and can survive without oxygen. If this thing is written can survive without oxygen, can we call them they are facultative anaerobes? Bit of facultative anaerobics and all those things you have to remember. The concept should be done. If not, I'm again and again telling, put me down in the comment section. I will make a short out of it so that you can always remember it. These are the pointers given about it. Okay, I have given you the most important pointers of it. These are the other pointers which you can go with it. Then they are what? Very important thing. They are called as jokers of plant kingdoms. Jokers of plant kingdom. Why jokers of plant kingdoms? Because they do not have their own shape. Why no shape? Because cell wall is not there. Always remember, beta, the cell wall gives a rigid structure to a cell, and they don't have any cell. They look like this. They could be of anything. Like this they are looking. Someday they are looking like this. Anything they could be there. So means they are joker. Joker don't have its own face. That thing is there. Now the major question, that how come they are joker of plant kingdom when they are present in the Monera kingdom? The answer lies to the question itself. That is, when there was earlier no class system present for Monera, means initially, okay, initially they were placed in plant kingdom. Remember in the last class I have told you what two kingdom classification. All the unicellular organisms were put into plants. Okay, they were placed in plant kingdoms. That's why they are called like this. So this is becomes what assertion and reason based question. Beta, it can come. Okay, now they are parasitic also. Normally they are saprophytic. Over here they are written that they are saprophyte and facultative. Oh, what happened? Okay. Now what is this? If you see that they are what saprophyte but facultative parasites. So they do what? Light blight of brinjal. They make also what? Broom, witch's broom disease. So means leaf change something like what? Like this broomstick. You know na? Broomstick is like this. So leaf changed like this in plant. This witch's broom, look over here, it is written in the potato and all. But this is what? Not only the plant disease, there are animal disease also they are causing. What kind of animal disease they do? They causes pneumonia. Even we can also be affected by it. So they causes disease both in the plant and the animals. It is not written in the notes. We are writing it like this. So two diseases, little leaf of disease of the brinjal and the witch's broom of the groundnut or potato. Groundnut is legume. Legume, what we have studied now. About legume, guys, we have studied that legume is what? Rhizobium is present with it as a nodule over there. So this thing you have to remember. Now the next thing is, this is with the Eubacteria. They are what? Photosynthetic. Okay, that we will see later on further, that you all know that what is their nutrition and everything. Then comes the Archaebacteria. Now they are what? They are the group of ancient bacteria which are very different. That's why Carl Woese did what? He put another domain for them itself. So they are the what? Oldest living fossils. They are obligate anaerobes. Means they do not require oxygens. They can survive in that area. What are those three types? Okay, that we will study. So before going to it, let's just see that how they are different from the other bacteria. Number one is that their cell wall, it is not made up of peptidoglycan. Means it is not made up of murin. It is made up of pseudomurin. It is what? Made up of pseudomurin, which is not peptidoglycan. Okay, it is more complex. Okay, so this thing is also there. Their cell wall is more complex. Okay, and they have a branched lipid chains. Normally the lipid chain is not branched, but here the branched lipid chain is there. Plus they have a enzymes, special enzymes which works at extreme conditions. Normally the enzymes work in narrow range of work, but their enzymes work in the extreme range, and this feature we're going to use it. So what are the special features which is making them survive in a hardest of environment? What is the hardest environment? Like Antarctica, they are present there. Like hot winds, like you might have seen that there are certain places, like if you went to Manali over there in the Kasol area, there is a Gurudwara and a temple over there. Okay, and it has hot water vents. Hot water is coming inside the hill, like inside the groundwater in the hill. So how come this hot water is coming? There also these kind of bacteria are present. Such 100 degrees Celsius, where if you put your hand into 100 degrees, you'll be like, ooh. But they are enjoying chilling, like chilling. Okay, so this thing is there. They're even present in the where you want to do like this. When you want to do like this, when we poop, or if a cow is putting a dunk and there is a cow area where a lot of dunk is there, you'll be like, what is this? A lot of people might have not seen it, but the people who lives in the rural areas, they might have experienced that thing, right? So they are present over there. So these becomes three different categories where they are present. Where they can be present at the hot water areas. They could be present at the Antarcticas. They could be present in the high salt area. How the salt will become a problem, we will learn in the kidney chapter very well about it. Then I will come back over here. Okay, so let's just see that what are the three types of this, and that will be the end of our topics. Okay, so that is what Archaebacteria can be present in what wait so we have methanogens, we have halophiles, and we have thermoacidophils. Okay, what are these? Let's just see that. There is what methanogens are, the methane producing one. What are the methane? Methane is CH4 beta. When I'm saying CH4 producing, who is producing? So when we fart, methane is present there. Especially high amount of methane is being released by the cattle and all, which is inflammable gases. So we use them for what? For cooking also. Okay, so for biogas productions. So basically what happened, the bacteria which is present in the swampy marshy areas, they convert carbon dioxide into the methane. Example remember is very easy because methano word is coming. Okay, so what are they? Methanobacteria, methanococcus. Two example you remember, if you can remember, methanomicrobium. Very easy. Methano word will come in all of these examples, so you can easily find it out. Examples easier is in this chapter if you put the tricks. So that's why I'm not even trying to give you the tricks over here. So these are what? These are important for the gobar gas fermentation, or we call it what? Biogas. Then an Archaebacteria is normally, this Archaebacteria is normally present where? In the gut of the rumen animals. That is what your cattle as a, because it helps in what? Digestion of cellulose. And later on it ferment it into the methane. So these things are present over here. So underlying thing you have to remember. In detail, we will learn how the biogas is working. Next is what? Is your halophiles. Okay, in your halophiles beta, okay, in halophiles, these are present in extremely salt condition. Halo word, philes means loving. Halo words is what? Salt. Now they are what? Purple membrane. In their pigment present, sorry, in their membrane, the purple color pigment is present. Okay, and they absorb what? Bright light and directly form ATP. Okay, but they are what? They directly form the ATP. Okay, now the example for this is what? Halococcus. You will remember at least one example which I am telling you. This one you have to remember. Then thermoacidophils. Thermo means temperature, high temperature. Acido means what? Low pH. Low pH means H positive concentration more. In this philes means loving. So they are present in the area where the temperature exceeds 80 degree and the medium is acidic. That pH should be close to 2. Okay, remember that our body, 1 pH is present for the acid in our body. We can't survive, our cell will start dying. But they are surviving over there. They are chilling. Okay, so they are present in hot sulfur springs and Himalayan regions. Examples of these are what? Thermoacidophils, thermoacidobacillus. Okay, so one such example that I'm giving you, which will be there later on also, what? That is thermoaquaticus. Okay, this is the bacteria. So if you remember that, how many bacteria we have studied in Kingdom Monera? 3 major types in our syllabus. One is what? Archaebacteria. Other is what? Blue-green algae, which is also called Eubacteria. And the one more we studied what? Mycoplasma. With this, the Monera kingdom is over. I know this is a longer videos and we should complete it more faster, but these things are important. I don't want any student who want to literally study should not have a resources which the coaching institutes provide us. I hope that you like this video. If you like this video, give it a like, share it with your friends who can't afford a expensive education, and come in the part of my mission that to reach out to the more students. We are coming up with many more things. For now, Kingdom Monera is over. For 11th class, next class would be on the upcoming Monday, which would be on the Protista, and we will be completing on the next Monday and Tuesday this chapter, where the 3 topics are left: Kingdom Monera, Kingdom Protista, Kingdom Fungi, and Virus, Viroid, and Brian's topic. I hope you are liking these videos. Well, that's all for this video. Thank you so much, and you will get the links for this notes and everything in the description box. Thank you so much.